Hi, this is a sciencey fellow here, and I'm here to teach mitosis today. So we all know that the process of meiosis in organisms is something that results in cells being formed that have half the number of chromosomes that you originally start with. And like one example of these cells, that are a result of meiosis are human gametes, which have 23 chromosomes. And each of these chromosomes, one sperm and one egg, they'll fuse to form a zygote which has that complete number of chromosomes. The process of meiosis is split into two parts, and we can think of these two parts as PMAT1 and PMAT2. So I'm going to take you through mitosis now. But before I take you through meiosis, just an extra warning before we start. The stages of before meiosis actually takes place, the cell goes through a process called interphase, which starts off by replicating the contents in the cells, including the DNA as well. So in humans with 46 chromosomes, interphase will duplicate these 46 chromosomes. So by the end of interphase, you're left with a cell containing 92 chromosomes. But really, to just make things much simpler, I'm gonna just illustrate meiosis using a cell that initially starts off with 4 chromosomes. And because interphase has taken place, the number of chromosomes after interphase will double, so that you now have 8 chromosomes at the start of meiosis. So there you go, the first stage of meiosis, PMAT1. The first stage of meiosis is prophase 1. And it's really an important part of meiosis. So really what's happening is that the DNA in the cell before this point isn't visible under a microscope. But actually, once prophase 1 has been reached, the chromatin condenses into chromosomes, and so the chromosomes become visible. And each of the chromosomes actually consists of sister chromatids. Parental and maternal pairs of chromosomes then pair up to form a bivalent, and the sister chromatids from one chromosome links at points called chiasmata, where sections of DNA are swapped, and this swapping of DNA is called crossing over. Then the nuclear envelope then breaks down, allowing the spindle fibers protruding from the centrioles, these little dots here really, to come into contact with the chromosomes for the next step of meiosis, which is metaphase 1. So at this point in metaphase 1, bivalents, they arrange themselves so that they're now at the middle or the equator of the cell, right here. So you can also call this area where bivalents line up as the metaphase plate. At the point of anaphase 1, each of the chromosomes in the bivalent move towards opposite ends of the cell. So they're separated and move towards the opposite poles of the cell. So note that chromatids that are making up each chromosome, they don't separate, it's just the chromosomes, not the chromatids, the 2 chromatids that make up the whole chromosome. It's the chromosomes. So both sets of chromosomes will end up reaching the opposite poles of the cell, and once this happens, nuclear envelope surrounds these 2 chromosomes, these 2 sets of chromosomes. And then after this happens, the nuclear envelope reforming around the chromosomes, the cell divides in a process called cytokinesis. And after cytokinesis takes place, it forms 2 daughter cells from the first original parent cell that we had when we first started meiosis. So PMAT2 is really the second part of meiosis. Just like a cake, when you cut it in half and there's one part and there's another part, this is the second part of meiosis. To help you remember, PMAT2. So first is prophase 2. As we kind of mentioned beforehand in our first half of meiosis, 2 two daughter cells are formed at the end of the first stage. But I'm just drawing one here to make it more simpler and adding a bracket times 2 to kind of show that this process actually happened to 2 cells rather than one that are just drawn right there. So in the process of prophase 2, like very, very similar to prophase 1, the nuclear envelope breaks down and spindle fibers form. So I haven't drawn the spindle fibers. The spindle fibers from the centrioles start to form. So there we go. Here's your centriole, and here are some spindle fibers forming from the centriole, ready to kind of make contact with the centromeres of these chromosomes. So at metaphase 2, so I've got a diagram here again. This represents that the metaphase 2 is happening to both the cells. The chromosomes arrange themselves so that they're the equator again, the middle of the cell. And again, like I mentioned before, the centrioles, the spindle fibers of the centrioles at both ends of the pole end up making contact with the centromeres. At anaphase 2, what happens is that the spindle fibers, they contract and they pull the sister chromatids now that make up each of the chromosomes to opposite parts of the cell, opposite poles of the cell. This really leaves you with 4 cells formed. They have a haploid number of chromosomes, which means they have half the chromosomes that you usually would have, or half the chromosomes that you started with in the first place. Crossing over has taken each of the 4 cells produced from meiosis are genetically different to each other, and also to the parent cell, the cell that you started off with when you first started meiosis.